day everyone, today I will be discussing the cell division. It is the mitosis and the meiosis. So, we have the mitosis, the division of somatic cells, the non-reproductive cells in the eukaryotic organism. So, a single cell divides into two identical daughter cells, and the daughter cells have the same number of chromosomes as the parent cell. So, next is the four subphases. The first, prophase, metaphase, anaphase, and the telophase, and of course to be followed by the cytokinesis. Looking for the move when cell is not dividing, DNA molecules is extended and condensed form is called the chromatin. Cell can only replicate and transcribe DNA when in extended state. When cell is preparing for division, DNA molecules condense to form chromosomes prior to division. Each chromosome is a single molecule of DNA and easier to sort and organize the replicated DNA into the daughter cells. Next is the prophase. Through major event, chromosome condense. The spindle fiber are specialized microtubules radiating out from centrioles. The chromosomes are captured by spindle. Next is the metaphase. The chromosomes align along equator of the cells with one kinetochore facing each pole. As you can see in the image, the centrioles pointing in the two dot above, and chromosomes is in the middle, and the spindle fiber is the line. And number 3 is anaphase. Sister chromatids separate. Spindle fiber attached to kinetochore shorten and pull chromatids towards the pole. And the first spindle fibers lengthen and push poles of the cell part apart. And next we have the telophase. The spindle fiber disintegrate. The nuclear envelopes form both groups of chromosomes. Chromosomes revert to their extended state, and cytokinesis occurs in closing each daughter nucleus into a separate cell. And next, and to be followed by the cytokinesis. The plants versus animal cell, and plant cells undergo the cytokinesis by forming a cell plate between the two daughter nuclei, and animal cells undergo to the cytokinesis through the formation of cleavage furrow. A ring of microtubule contract, pinching the cell in half. So we will proceed to the meiosis. It is a single cell divides into four unique daughter cells. The daughter cells have half the number of chromosomes as parent cells, so they considered haploid. We have the interphase, meiosis 1, meiosis 2, the daughter nuclei, and the daughter nuclei 2. This one, the homologous chromosome. Next is the genetics terminology. Terminology are applied. It refers to the number of set of chromosomes in cell. We have the haploid or the one copy of each chromosome designate as N, the number of chromosomes in one set. Gametes. Diploid, two sets of chromosomes, two of each chromosome, designate into N in somatic cells. The diploid organism receive one of each type of chromosome from female parent or maternal chromosomes, and one of each type of chromosome from male parent or paternal chromosomes. Next is the genetics terminology of homologous. Chromosomes exist in homologous pairs in diploid 2N cells. The exception is the sex chromosomes or X and Y, other chromosomes known also as autosomes. They have the homologous. Next in here, the sexual reproduction. Fusion of two gametes to produce single zygote and introduce greater genetic variation allows genetic recombination. With the exception of self-fertilizing organisms, zygote has gametes from two different parents. Next is the sexual reproduction in humans. At fertilization, 22 chromosomes are donated by each parent, the total of 46 or 20 pairs. Gametes are the sperm or ova, contain 22 autosomes and 1 sex chromosomes, are haploid and haploid number N is equal to 23 in humans. In fertilization result in diploid zygote. The diploid cell in Qn equals to 46. N equals 23 in humans. Meiosis, sex cell or gamete formation. In meiosis, there are two divisions of nucleus, the meiosis 1 and the meiosis 2. We have here the meiosis and the sexual reproduction life cycle. So the next is the genetic variation in diploid organism. The fusion of sperm and egg result in unique offspring, but not only because the young are the product of two individuals with different genetic makeup. The meiosis shuffles the genes so that the individual's gametes are genetically different from one another. So is the shuffling accomplished? Shuffling of meiosis 1 in addition to a new combination of chromosomes resulting from fertilization. There are also events in meiosis 1 that shuffles the genes. Number 1 is the crossing over in prophase 1 and independent assortment, and number 2 in metaphase 1. Crossing over or the homologous break at identical location that rejoin opposite partner. These recreate the combination of the alleles on each chromosomes. Occurs randomly several times on every chromosome, result in mixing of genes you inherited from your parents. Male produces sperm throughout life after the onset of puberty, about 1,500 sperm per second. Oogenesis in female is probably complete either before or shortly after birth. During oogenesis, three polar bodies develop as the mature ovum is generated. Polar bodies contain the little cytoplasm and eventually degenerate. The primary primary oocyte communication, second oocyte, first polar body, mature ovum, and the polar bodies. Now is indicated here the image of the mitosis and the meiosis cycle.